everyone, welcome to my channel Biswa's Coaching, and my name is Biswa Choudhury, your online instructor. So today I am going to discuss about all the very related but very confusing terms of chromosomes, like what is chromatid, what is sister chromatid, what is chromosome, what is haploid chromosome, what is diploid chromosome, what is homozygous, what is heterozygous, what is homologous chromosome. These are very confusing but very important terms. So let's start today's topic, that is called chromosome. So chromosome, the term came from the Greek words, that is chroma means color and zoma means body. So color, body, as you can stain this thing under a microscope, you can stain and observe this thing under an optical microscope. So that is why it is called chromosome. So we know all the structure of a chromosome. It is something like this. There is a constriction, and after that there is a gain. I think you know this, the structure is something like this, but I have created the drawing something like that because I just want to show you guys, because the DNA is super coiled inside the chromosome. So DNA is coiled-like structure inside the chromosome. That is why I have drawn something like this. So each chromosome contains a double-stranded DNA. So each chromosome made up of double-stranded DNA. Okay. So if I consider, suppose I am considering this thing, there is small constriction, not exactly at the center but towards the center. That is why one arm is smaller than another arm, because this arm a bit larger and this arm a bit smaller. But you cannot say this is chromatid. You cannot say that. This is chromosomal arm. One arm is called P arm and another arm is called Q arm. So which one is P and which one is Q? The larger one is called Q and smaller one is called P. The easiest way to remember this thing, suppose just write from A to Q, A to P. Okay, A, B, C up to P. Now again write A to Q. I am including another alphabet for QR, and this is called PR. Okay, so now just draw a length including this alphabets and draw another length including Q also. So as it is very common that the Q comes after P, that is why this arm is obviously larger than this one, this arm. So it is better to remember this thing by drawing the alphabets. Okay, so this arm is larger and this arm is smaller. Okay, so when should I say that which one is, why it is not called chromatid and why it is called chromatid? Chromatid is only called in a chromosome when this chromosome makes another copy of it. This chromosome makes another copy of it. When you can get this kind of structure? After DNA replication. So it is not always DNA is replicated inside our cells, because DNA is replicated when the cell needs to be divided. Okay, so when the cell is in the interphase phase, so that phase is called interphase. When cell is at the interphase, then DNA is not replicating and cell is not dividing. When the cell starts dividing, prior to that DNA is replicated. Okay, so interphase chromosome, if you can see the interphase chromosome, then you cannot distinguish which one is chromosome 1 and which one is chromosome 2 and all. You cannot distinguish these things, because during the interphase, chromosome structures look something like this. Suppose this is chromosome 1 and this one is chromosome 2. They are noodle-like or thread-like structure. That is why it is called chromosome. It's a thread-like structure. Sometimes we refer this phase of chromosome as chromatin. Chromatin. There is another definition of chromatin when I will talk about DNA packaging, but if you say this phase of chromosome is chromatin, that is also correct. Okay, based on this term we can distinguish which one is euchromatin and which one is heterochromatin. So that is why it is called chromatin. I will talk about euchromatin and heterochromatin in next class or in some future class, near future class. Okay, so this is interphase and this is called chromatin. But when after these chromosomes are get replicated after this interphase, then the chromosome will make another copy of its own. Okay, so it is holded by the centromere position. The replicated chromosome, just after the replication, it is not separated from each other. Okay, so if I consider this is chromosome 21, this is another chromosome 21. So one arm, so one half is called, if I consider, if I make a division from the middle, then one half is called chromatid and another half is called also a chromatid. Okay, so after replication you can say that this half is called chromatid and this half is called chromatid. This is another chromatid and holded by the structure called centromere. This one is called centromere. If I consider the two chromatids at once, then we can say these are sister chromatids. They are sister chromatids. Why they are sister? Because they are identical. They are actually identical. That is why they are called sister to each other. Chromosome will get packed and forms the structure something like this during cell division. But not prior to that. During interphase, if I consider the interphase chromosome inside the nucleus of a cell, if I consider this is cell, this is nucleus, and these are chromosomes. But during this chromosomal interphase, the chromosomes are not replicated, but as they are loosely bound, they are not condensedly bound, they are loosely bound. That is why they are actually involved in transcription and translation, because they are loosely bound. That means the RNA polymerase and all the transcription factors are allowed to come here and access the DNA structure. So that is why they are involved in transcription and translation. Translation. So you can see DNA replication is not always happening. DNA replication only happens before cell division. This is very obvious because cell is not dividing all the time. But during the development of our body, the cell is dividing. Okay, but transcription and translation is very important to maintain our life, because after this translation, protein is produced. That protein is utilized by our body. So that is very important to maintain our life. Okay, so during the interphase, transcription translation happens. But after this interphase, they make another copy of its own structure, the chromosomal structure, and they start condensing. After this condensing, they look something like this, and this is called chromatid, and both of them are called sisters to each other. Okay, so these are, and this is called, this constricted structure is called centromere. And why they are holded by the centromere? Because there is a special kind of protein called cohesin present in the centromere position, which allowed the two chromatid holded to get bonded. Okay, human contains 23 pairs of chromosome. Human contains 23 pairs of chromosomes. Okay, among them 22 pairs is called autosome and 1 pair is called allosome. Allosome is also called sex chromosome. Okay, so if I consider only the 22 pairs of autosome, then their nomenclature is from chromosome 1 to chromosome 22. Chromosome 1 to 22. And their size is also very important. Their size is also very important. Like chromosome 1 is the largest one, and chromosome 22 is the smallest one. So based on their numbering, if I consider the length is arranged in descending order. The length is arranged in descending order. Like chromosome 1 is the largest chromosome, chromosome 2 is the second largest chromosome, chromosome 21 is the second smallest chromosome, and chromosome 22 is the smallest chromosome. Okay. Suppose this is chromosome 20, suppose this is chromosome 1. So, one pair of chromosomes, 1. That means this is another chromosome, 1. Now this is chromosome 2, this is another chromosome 2. Now this is chromosome 3, this is another chromosome 3. Okay, in this manner, I consider this is chromosome 21, and this is chromosome 22, and this is another chromosome 22. That means this is one pair, okay. Sorry. This is another pair, one pair. This is chromosome 1, this is chromosome 2, this is chromosome 3, one pair. This is another pair, one pair. This is chromosome 22. Okay. I am just taking the autosomal part. Okay. Now, here one pair of chromosome. Just suppose this is interphase chromosome. Okay. So 22 pairs means 44 chromosomes. 44 chromatid. I cannot say this is chromatid because they are not folded. Okay. When I can say this is chromatid? After. So I can say this is 44 chromosomes. It's better to say 44 chromosomes. It's not chromatid. Okay. Now, DNA is replicating. Okay. When the DNA is replicating, that means you can get after replication not just one pair of chromosome 1, but two pairs of chromosome 1 after this replication. This is another pair, this is another pair, this is another pair. Okay. This is also another pair, this is also another pair. So instead of getting only 44 chromosomes, you are getting 88 number of chromosomes. 88 number of chromosomes because they are replicated. Okay. Now they are called, these are sisters. These are sister chromatid. These are sister chromatid. Okay. These are all sister chromatid. But how can the chromosome number is maintained in a cell? So after this replication, one cell is got divided into two cells. That means one of them separated in one cell and another one of them is separated and taken by another cell. Okay. Suppose I am taking this part only. It is present inside one cell, the nucleus of a cell, but I am considering only one cell. Now, during cell division, they are separated. Okay. So, if I consider the separation, one cell get one copy. So that means one cell get chromatid, one chromatid of chromosome 1, and another cell get another copy of this chromatid. Okay. Likewise, one cell get one copy of this chromatid, and another cell get another copy of its chromatid. So this is one cell and this is another cell, because they are separated during cell division. They are separated. So one cell contains one chromatid, another cell contains another chromatid. Likewise, this cell, this chromosome 1 also gets separated, and one cell contains one chromatid, another cell contains another chromatid. So you can assume that this is now the chromosome number is maintained. Okay, so you need to get only one pair of chromosome in each cell, but before cell division they have got one cell is getting two pairs, but that is not maintained in a cell. So how the cell can maintain the number of a chromosome? By dividing the cell the chromosome numbers. So one cell is getting one pair, another cell is getting another pair. Okay. So now, as they are same chromosome, this is chromosome 1, this is chromosome 1, this is chromosome 1, this is chromosome 1, but they are not sisters. These are not sisters, but these are sisters. Okay. Likewise, these are sisters, but these are not sisters. So what are they? They are actually homologous chromosome. They are actually homologous chromosome. I think now it is clear to you. This is very easy. What is haploid chromosome? You know, haploid, if I consider only the one chromosome, this is called haploid. Just for your quick recap, and this is denoted by N. That means 1, here N equal to 1, that means one chromosome. And now diploid means when I am considering another copy of its own, this is called diploid. That means 2N, double the number. So 2 into N is what? 1 equal to 2. What is homozygous and what is heterozygous? For that you need to understand that one chromosome contains DNA, and within this DNA part, some parts are gene. Gene means which actually produce some functional molecules. I will talk about this gene in my next class later. So now you need to remember the genic location. Suppose this is chromosome 1 and another chromosome of, suppose this is also chromosome 1. Okay, if there is one gene is present here in the chromosome, so obviously there is another gene is present just to the next chromosome. If there is another gene is present here, another gene is present here, because they are homologous. Homologous chromosome means they are actually getting the same copy of a chromosome, that is chromosome number 1, but maybe their forms are different. I am coming to that part. So gene is present here. So one human body contains two forms of a gene. one form is present in this chromosome, another form or variation is present in this chromosome. Suppose this is for gene for tallness. This is responsible for our growth. So, if there is one gene is present for tallness, obviously there is another gene is present for tallness because two chromosomes. Now, if they are, so these are called alleles. So, allele is called what? Allele is called two forms of a gene in a homologous chromosome. If I consider the homologous chromosome, there is two forms of a gene. Why I am telling this is two forms? Maybe this is responsible for tallness. Maybe it is when it is expressed, it produces tallness. Better to say this: the gene is called growth. For growth, okay. Gene regulating the growth of our body. So maybe if it is expressed, it causes tallness. But if it is expressed, it causes dwarfness. Dwarfness. So which gene will get expressed? That is determined by some cellular programmings and all. I will come to that part when I will discuss about epigenetic part. Okay. So this is different forms of growth. One is dwarfness, one is tallness. So if one homologous chromosome contains two varieties of a gene, they are called heterozygous. Heterozygous. But if one homologous chromosome contains same form of a gene, like instead of dwarfness, if there is another tallness, then it is called homozygous. Homozygous. If I consider X chromosome, suppose this is X chromosome, in human body, in male, suppose this male, there is no another X chromosome is present. Instead of that, there is one Y chromosome is present. Suppose this is Y chromosome. This is X chromosome, this is Y chromosome. In male body, there is one X and one Y, but there is no two X. So there is one form of a gene present, but there is no such counterparts because they are not homologous. X and Y are not homologous chromosomes because their size, shape, their gene orientations are different. When I consider only chromosome 1 or chromosome 2, they are homologous because they are identical. Their size, shapes are identical. But X and Y are not homologous. So, the gene present in the X chromosome, there is no such other form present in the Y chromosome. So, this kind of thing is called hemizygous. Hemizygous. Okay. Hemi means only one form is present instead of two forms. So, you have now the basic idea what is hemizygous, what is homozygous, what is heterozygous, what is homologous chromosome, and how the chromosome number is maintained in a cell after cellular division. These are very clear to you, and what is chromatid, and you also know what is P arm, what is Q arm, how can you remember this P and Q arm. Okay, so if you like this tutorial, please hit the like button, subscribe to this channel, okay, and don't forget to share it with your friend, and also commenting down if you have any suggestions. Okay, thank you. Thanks for now.